So good morning, everyone. First of all, I would like to thank David for the invitation. It's really an honor to participate in the symposium with this amazing list of speakers and attendees. So thank you so much. I also think that the topic of the meeting is very appropriate because given the recent explosion of interest in generative AI that is making people think or ask whether there could be cognition outside brains in non-living systems, in machines. And I think before we even start asking this question, we should start thinking whether there can be cognition outside brains in living systems. And that's why we are talking about cellular cognition, for instance, in this meeting. And I'm aware that there will be quite a few talks in this topic. And I'm really looking very much to them. So we have already seen a few minutes ago very nice examples or a very nice description of how the brain is able to process information coming from its environment. And this is a non-trivial task because this information is frequently complex in time and multidimensional. It comes from many sensory modalities that the brain has to integrate. This is not at all a trivial task. But I would like to claim, and I think most of us will agree in this audience, that the same problems are faced by cells, by single cells. They have multiple signals coming from all their signaling systems, and these signals in the real world are definitely not stationary. They are changing all the time, and the cells need to be able to respond to them. Here, for instance, you can see an example of early embryogenesis in mice. This is an experiment by Kate McDowell published a few years ago. And in this example, every one of these cells, if you look at each one of these cells is faced with its own environment, which changes continuously in time, different from one cell to another and different from time to time. And I guess we can all agree that the outcome of this developmental process is rather reliable. And yet, I guess we agree that this cannot be programmed in the cells to begin with. So the cells need to be able to respond to their environment. Each one of these cells, which are genetically identical or almost identical, they need to be able to respond as time moves on and processes information. And the question is, how do the cells do it? So how is the cell doing this type of processes? So to try to address this question, maybe we can go back to our analogy and think back of how brains do it. And going a little bit in a different direction from what Professor Friston has really told us about, I would really like to claim that something that is, of course, very well known, that the brain is doing this type of computations through neural networks, right? And of course, there is a very evident analogy between neurons and cells, because cells are also driven by networks of dynamic elements, which are genes. So the claim would be that the same way that neural networks are able to do this kind of complex information processing, we can also think in terms of complex information processing that could arise through this gene regulatory network, for instance. In general, this is nothing new. It's well known that networks of dynamical elements can perform this type of information tasks. This has been, of course, co-opted by computer scientists for many years in the field of artificial neural networks. And we know that specific architectures, such as, for instance, this multi-layer feed-forward network that you see here in the screen, is very well suited to process and classify information of different sorts. Interestingly, from my point of view, the models that the computer scientists and machine learning researchers use to describe these networks is, in my opinion, better suited to describe the dynamics of gene regulatory networks than neural networks. To begin with, neurons have spikes. Neurons are spiking elements, and this is completely ignored usually in these models. So in some sense, the expression artificial neural network could be a misnomer. In fact, maybe I would claim that it would be more appropriate to call them artificial genetic networks in that regard. Okay. So the question is, well, so far so good. This is very conceptual. Is this really what happens? Well, the good news is that we have a lot of information from public databases about these networks that we have inside cells. We can take a look at any of these databases. For instance, this is EcoSeq, which is very well-known and has been around for many, many years and updated continuously about all the regulatory interactions that you can see in E. coli. For instance, in this instance that we analyzed some time ago, we have more than 3,000 genes connected by more than 8,000 edges. And the question is, what is the architectures of these networks? So we can analyze them. This is a very simple thing to do, and I will tell you in a minute how we did it, even though it's very simple. But let me tell you the result. If you take a look at these more than 3,000 genes and look at how they are structured, what you see, and this is data and information is available, they are structured in this multi-layer feed-forward manner. Most of the genes, almost all 3,000 genes are structured in this manner, which is very different from what you would get, I would claim, if you obtain a network like this by chance. Okay? So hopefully this is something that the cells are using. So maybe this would be the end of the story. That's it. We have beautiful information processing systems because cells are using this kind of deep multi-layer feed-forward networks at the level of gene regulation. But of course, as we all know, and it's very clear, this type of networks have certain limitations. First of all, they have no memory. These feed-forward networks have no memory of past events, okay? Because information is flowing from one side to the other, and once the information goes through it, the information is gone. And the second thing is that these networks do not scale. The information processing capabilities in the network don't scale with additional tasks. If I add another task, I have to recalculate all the weights. So this is something that is well known. So how do we solve this problem? And of course, people, computer scientists, computational neuroscientists, for many years have been addressing these issues. And one way of solving at least the problem of memory is by going from feed-forward networks in which information is progressing from left to right and it's gone, to recurrent networks in which information persists for a while, and then this gives some sort of fading memory to the system, which enables the network to integrate information from the environment in a continuous manner. Okay? So that's something that has been around for many years, and in the last 2 decades, for instance, there have been multiple proposals going in this direction, mostly within the field of computational neuroscience, a little bit also in machine learning, in the idea that we have this kind of recurrent networks that receive information, they project this information in a multidimensional, nonlinear multidimensional way, and this provides a system with strong information processing capabilities. Of course, the issue then is how you do train the network, and and a very simple way of doing it is that, for instance, what people like Herbert Jäger and Wolfgang Maass showed is that you only need to train the output layer, the weights go into the output layer, and this means that this is at least a feasible problem, a feasible solution to the problem, okay? This has been, we can call this, and this has been known in the field as reservoir computing, because what the information processing system is basically a recurrent reservoir, we can call it like this, which receives the input, projects it into this multidimensional space, and then you have a bunch of nodes, which could be neurons, or I would like to claim that they are genes. And these nodes, basically what they do is they interpret the high-dimensional dynamics of this recurrent network. So that's the idea. Now, the advantages of this type of architecture, as I was saying, is that they can process, of course, distributed inputs, multiple inputs, because this is what neural networks do. They can also process temporal information, as I was telling you, because the information remains for a while, and they are relatively easy to train, depending on the scheme. And there are few schemes, not only the one that was proposed by Jäger and Maass. So then the question is, well, I just told you that most of the genes in the transcriptional network of E. coli are distributed in this feed-forward manner. So what? Is there any recurrence at all? Well, how do we look for recurrence? And this is very simplistic. I'm sorry for being a little bit simple in what I'm going to tell you, but what you can do is something very easy. So you have your network here, and you want to find out how many recurrent, if there is any recurrence in this network. So one thing that you can do iteratively is, first of all, you remove all the nodes that are upstream, and you can do because you remove the nodes that have no inputs, then you remove the nodes that have no outputs, and you do it iteratively, and eventually, when you cannot remove any more nodes, the nodes that remain are connected all to all in a recurrent manner. There is nothing downstream, there is nothing upstream, everything in the middle is a recurrent reservoir, what we would call. So we can apply this extremely simple procedure to all the databases that we have, and what we see, for instance, in the case of E. coli, is this. So you have a subset, a relatively small subset of genes, upstream of this highly feed-forward layer, and this subset is basically a recurrent reservoir that could be performing this kind of multidimensional projection in time of the inputs. Okay, so that's what we see. we analyze this for many different species, not only E. coli. We can use it to many different databases from yeast, including yeast, fly, and humans. And we get a collection of reservoirs. So the structure is similar in all these cases. We have upstream a relatively small reservoir. The case of Bacillus is not very representative because the network was not very large, the one that we looked at the time. But you see a small number of genes which are feeding upon a relatively complex feed-forward architecture that basically what it would be doing is would be interpreting the dynamics that is seen in the recurrent core. So how do we test this? So what we did in the past, and I'm not going to, I don't know how, I hope I'm not going to, I forgot to start. Okay, good. So what we did in the past is we started being very, very abstract. We were not very biologically realistic. We chose this architecture, this network that we have obtained from these databases, and applied all the techniques that the machine learning researchers use to see whether the networks work well and to validate them. For instance, one of the things that we can do, first of all, is how do we train the network? And we can train the network with a very simple linear algebra approach, in which once we have the time-dependent dynamics, we apply this type of rule, for instance, a linear ridge regression in this particular case, and we obtain the weights that go from the reservoir to the readout. And this means that for this particular task, the readout is going to be the interpreter of whatever is going on in the reservoir. That's the idea. Okay, that's one thing. And the second thing is once we have trained the network, then what is the task? So again, this is a very non-biological task, but this is a task that people use in the machine learning field, in the case of reservoir computing or recurrent networks. For instance, the idea would be we introduce some kind of input. I really don't care so much, so the input could be random even, and that's what we have here. This input is driving this network. And then what we want to see is whether the network is able to replicate a very complicated transformation. Okay, so we take this input and we want to transform it in this way, which is rather terrible, but the idea is that you have nonlinearities here, and also importantly, you have to remember past states. Okay, in this particular case, the previous 9 steps in the iterative process. Okay, so what it means is that we have a function which is highly nonlinear and that requires the system to have memory. So it's a very stringent condition, and the question is, do these networks, which we know that exist because we got from the literature, are they able to reproduce this complicated function, which is way more than I think a cell is going to have to do? So we are being very stringent here. And for instance, I'm going to tell you very simple results. Here, this would be an input, which is a complicated input. Nothing is basically random. And then this is the function that we want to reproduce, so the transformation between input and output. And we want the system, the reservoir, to produce this output that you see in black here. And indeed, what we can see after training, this is after training, okay? So we train the network, we train the weights, and then we let it go, and we apply an input again for the same fixed weights. And we can see that indeed the network is able to reproduce this very complicated nonlinear and memory-driven function. Of course, this is very artificial, so the next thing that we did, and then let's see if this works, because now it's not going. Oh, there we go. Oh, there we go. So yeah, so here you can see that the same thing can happen with the other species. Of course, that's nothing very surprising. And then we start adding biologically reasonable situations. For instance, we started including, we know, of course, because this is a published database, so we know exactly which nodes, which genes in the reservoir are receiving which type of information. So we can have these relatively biologically realistic inputs. And we see that the error, this is the normalized root mean square error here, is basically the same for the different types of inputs from the same error that you would have if the inputs were completely random, and the same number of inputs that you can see in the line here. This would be corresponding to random inputs, okay? So this works for biologically realistic inputs. And then the other thing that you might think, okay, so how biologically relevant is that we train this network with a ridge regression? Are cells doing ridge regression? Of course, the claim is not. But then what we can do is substitute the training procedure, for instance, by an evolutionary method. Okay, so of course we can use genetic algorithms that, of course, computer scientists have developed over the years. But in this particular case, the claim is that this is closer to what biology would be doing. Evolution might be training these weights. And indeed, this is working, because if we use an evolutionary approach to train these weights, we see that after a few thousand of generations in this particular very complicated task, we get to the same kind of error. So we can train this network by means of natural selection, if you will. Okay, so in summary, so far, what we have is a situation in which we have a complicated input here, constantly changing in time, and we have an internal model similar to what Professor Freestone also was talking about, more or less. The cell has an internal model of the world, which is basically the high-dimensional dynamics of this reservoir. That's the claim. Okay? This dynamics of the reservoir is a dynamical model of the world that the system keeps updating with the information, and then it's going to be able to reproduce a relatively complicated output. That's the claim. Now, I don't know if I have 5 more minutes or less. I don't know. 5 minutes. Okay, so in the last 5 minutes, I would like to discuss another aspect, in my opinion, that relates to cognition of these systems, which would be the idea of the memory effect, okay? So I just told you a few minutes ago that this recurrent reservoir is providing some kind of fading memory. So the memory is limited because any input that enters into a reservoir eventually is going to decay. It's going to decay because there is dissipation, so this is going to decay. So if you inject a pulse, after a while it's going to go down. Of course, we are not injecting a pulse, we are injecting a continuous system, but the fact of the matter is that in principle this system exhibits short-term memory. So the natural claim would be, what if I want long-term memory? Well, our solution would be, well, the long-term memory would be hard-coded somewhere else in the brain, for instance. But what I would like to say is that this paradigm allows us also not to have to hard-code the long-term memory. Okay? Also, the dynamics of this system could encode both for the short-term memory and the long-term memory. And how can this happen? Well, in order to do that, we have to consider a situation in which this reservoir has a self-sustained dynamics. I just told you that if you inject a pulse, the pulse is going to reverberate and decay. But of course, a network like this, we know very well, networks of this kind can have self-sustained dynamics. That doesn't need to be regular. For instance, in this particular case, it would be chaotic dynamics. So going back to computational neuroscience, and I'm going to move now to neuronal networks for the rest of the talk. The idea is that we know for many years that in the case of neural networks, which have a balance of excitation and inhibition, these networks very naturally go into a chaotic state, usually high-dimensional chaotic state. So far, so good. So this means that the networks can have a self-sustained dynamics in the absence of any input. Why is this good for? Well, there is something interesting of chaotic systems. And this is exemplified in this slide that I have here. Chaotic systems have a property which is called generalized synchronization. This is a very dark, a very cryptic name, but let me tell you what it is. Okay? What is generalized synchronization? If you have a chaotic system, and I'm sorry because now I'm changing also, I'm going to a different experimental setup because this is the one in which this thing was reported experimentally quite a few years ago. Imagine that you have an optical system, a laser system that is intrinsically chaotic. This is an example, this is just an example of a chaotic system, like our neural networks are, okay? So I have a system which is here in this box, which is intrinsically chaotic. Now, imagine that I drive this system with this complicated input here. Okay, which in principle is not related with the intrinsic dynamics of the system, okay? But I drive it. So generalized synchronization of chaos means that every time I drive the system with the same drive, one time, another time, the system is always going to respond in the same manner. You might say, oh, this is trivial. Well, this is trivial if we are talking about a pendulum. This is trivial if we are talking about a system with regular dynamics. If we are talking about a chaotic system, you know that chaos implies sensitive dependence on initial conditions. So one might think, oh, every time I drive my chaotic system with the same driving, I'm going to get a different result. But that's not the case. Okay, this is a property of chaotic dynamics, that you have this kind of generalized chaos. So every time I drive it, the system responds more or less in the same manner. These are the two responses. These are experiments. The two responses after you subject the chaotic system to the same driving. Now, I would like to claim that this is sort of a long-term memory, because what is a long-term memory? A long-term memory is that your system is going to respond, you are going to respond always in the same. If you remember a movie that you saw, I don't know if it happens to you, but every time I see a movie that is a drama or something, I always cry in the same places. No? So, so my brain is a complex system. I have a complex information, and I'm always responding to the same stimulus in the same way. This is for me, and this is dying. This is for me an example of this generalized chaos synchronization and long-term memory. I think this has died. Let me just, oops, I have batteries here. Sorry. Oh no, it works. Okay, it works. Okay, good. Okay, so now let me, in 2 minutes, let me tell you that these networks, this type of paradigm also provides you with the same type of behavior as long as the reservoir is operating in a chaotic regime. And to do that, instead of going into a cell, I'm going into a minimal brain, if you will. So we were talking about minimal determinants. I'm going into a minimal brain, which is that of C. elegans with 300 neurons. We know, of course, very well how it is connected because the wiring is very well known. So we can do the same analysis I told you earlier about how is this neural network structured. And the neural network is structured in the same manner. So you have this reservoir, a bit larger than in the case of E. coli, driving all these neurons downstream. So what we did is we took experimental data from the lab of Venkatachalam. At the time he was at MIT, I think, and they were measuring the dynamics of a few neurons. They had the dynamics of a large number of neurons, but we only knew 8 of them. That we could only identify with 8 of them in our model. So we use this data and we use a genetic algorithm, in this case just for purely practical reasons, to reproduce the network. One of the things that we know is exactly the connectivity. But what we don't know in these databases is which connections are excitable and excitatory and which connections are inhibitory. So what we did is, in order to really have a network that mimic the behavior of the worm, basically we applied a genetic algorithm to identify whether the connections were excitatory and inhibitory. And as I told you a minute ago, this is very important because this balanced excitation inhibition is crucial for this system to exhibit this kind of self-sustained chaotic dynamics. Okay, so we obtain a fit. This is basically the genetic algorithm as it progresses. This is the fitness, and we can see that at the end, the best fit that we get is for a situation in which we have more or less a good balance between excitation and inhibition. Almost 50% of the synapses are inhibitory. Okay, and by the way, and this corresponds to a situation in which the complexity of the signal is maximum. Okay. So we have maximum complexity when we feed the experimental data for a situation of balanced excitation and inhibition. So this means that our reservoir has a self-sustained complex dynamics. So now the question is, do we have generalized synchronization? Well, what we can do is we can apply the same input. In this particular case, it's not a movie, it's just a train of square pulses that you can see here, that they are not periodic. The separation varies randomly, if you will, so it's a complex input. And then what we do is we apply the same input to our network once we have adjusted it over and over and over again, and we look at the different trials. And we compare the response of the system to the different trials, and as a control, we compare with the response to different pulses within the same trial. So basically we have a comparison of the inter-series correlation with the intra-series correlation. Okay? And what we see is not spectacular because we want to claim that this is biologically realistic, but we see is that the distribution of correlations is higher for the inter-series and for the intra-series comparison, which means that the system responds in a more or less reliable and consistent manner to different inputs more than it responds as time proceeds. Okay? And we can quantify the reliability of the system by looking at the cumulative distribution of correlations and the area under the curve here. So we use this area as a parameter that measures the reliability. And indeed what we see is that the reliability in our optimal case, in which we have balanced excitation and inhibition, is really higher than 0. 0 here, it would mean that the intra-series and the inter-series correlations are the same. This would mean that really the system is not responding reliably. It has no long-term memory. Here, the fact that this distribution is clearly different from 0, this means that consistently the system responds more or less in the same way to multiple repeated presentations of the input, and therefore we could claim that this is some sort of long-term memory which is not hard-coded. It is soft-coded in the network. There's no specific module. It's just the dynamics. The dynamics enables for short-term memory and long-term memory, provided the system has intrinsic dynamics. And with that, I am going to stop. This is a summary. I have shown you that gene regulatory networks and biological networks in general have a recurrent core, that this network architecture enables reservoir-based computation, and these complex time-varying inputs are integrated as time proceeds in an online manner in the dynamic state of the networks. That's it. Thank you so much for your attention.
Thank you. Fascinating. I have a question. If the network, the reservoir operates in the chaotic regime, would that mean that you can, I mean, both the long-term and the short-term memory will be present for the same chaotic regime? Because the first description of the reservoirs was that they would operate at the edge of chaos, not in the chaotic regime.
The paradigm, when we were looking at the first tests, we were in the our networks were forced to obey the echo state property, which, as you say, this means that they are not fully chaotic. If you ask me whether when the neurons, when the network is operating in the fully chaotic regime, I would still like to claim. Of course, it's harder to measure, but I would like to claim that there is still short-term memory.
But is it possible to test it with some task?
Yeah, we haven't done it, and it should be possible. Yeah, that's a wonderful question. Yeah. We have. I mean, unfortunately, the two parts of the talk are in slightly different regimes, but we haven't done it. Yeah, definitely. Thank you. Thank you so much, Anita.
Go ahead, please.
Hi, Joel Mefford, UCLA. Nice talk.
Thank you.
So for your empirical analyses, like when you estimate the recurrent core of a network from, say, the bacterial transcription data, first you have to identify the edge set, and that can be done with false positives and false negatives. And in particular, it's probably biased towards like highly expressed genes because you have lower power and higher false negative rates for rare genes. So how stable are your conclusions about the computational capacity of these networks or the biological plausibility when they're estimated with error and probably biased?
Mm-hmm. Okay, I guess the answer is going to depend on what type of data we are using to establish the network, right? I mean, I'm not sure if you are referring, because one thing that we are doing more recently is there is of course a lot of single cell RNA-seq data that are available out there, in which at least we know the abundances of these genes and the correlations between them. These data sets are very noisy, and I'm not sure, as you're saying, we would bias the information given the average abundances. And this is a problem that we are encountering. As far as the databases that we are using are based on structural information of DNA binding to transcription factors. So I'm sure that there's going to be some bias, but what I can tell you is that we have been analyzing the system as the years have progressed, and we don't see that there is large changes in the structure, in the architecture of the network, just because more transcription factors are being found. I'm not sure. Maybe later you can tell me if you think there's going to be large biases in. I mean, we are not using using data that depends on the abundance of the different elements in such a way.
Maybe I'll follow up later. I was more specifically wondering about the stability or the robustness of the computational capacity. When the error is when the core network is estimated with error for whatever reason. Yeah, definitely. And thank you so much. One thing that we can say is that if the network is if the recurrent core is very small, eventually it's going to lose its computational capabilities. But we have done analysis. I haven't shown you the data, and definitely the computation is robust. And it's also, I have to tell you, the best reservoirs are random networks. We are not using random networks at the advanced. So if the network is random, it's going to perform. Usually, I mean, it's the best way that these things perform. Our networks, which are not random, perform very well. The computation is very robust. It's very robust to the architecture. Yeah.
Okay, I think we'll have
to end
it there. Thank you very much.
Thank you so much.